Okay, it's all yours.
Good morning, everyone. My name is Rahul. This is David. We're both from McGill University in Montreal. So basically, based on the case study, our approach towards this ependymoma case is to target aquaporins 1 and 4 to prevent the migration and invasion of the infratentorial ependymoma after surgical resection.
So a bit of background on ependymoma. It's a highly variable primary CNS tumor that arises in the ependymoma, which is the neuroepithelial lining of the CNS. So the cause is unknown right now, so there's no concrete treatment available. The current treatments are based on symptoms to treat the symptoms that arise, and there are actually a couple of clinical trials that are in place right now, but nothing concrete has come out of it. The overall 5-year survival is pretty good at 83%, but that's lumped up all of the subtypes of ependymoma, not the specific case we're going to be talking about today. So Xavier's tumor, as you see in the MRI here, was present in the fourth ventricle of the posterior fossa, which classifies it as an infratentorial ependymoma. The current treatment is basically the classical treatment for any brain tumor. You surgically resect the tumor, and either treat it with radiation or chemotherapy, or a combination of both to avoid the chance of any recurrence. Now, our approach is based on research that has shown a natural abundance of the Aquaporin 1 and Aquaporin 4 water channel proteins in infratentorial ependymoma. And there's also a very important clinical significance. Overexpression has been found to cause migration and invasion of tumor cells. So what we want to do is that we want to directly inhibit this with small molecules such as AQB013, and this has been shown in the colon cancer adenocarcinoma model that actually reduces the invasion and the migration of the tumor cells. So a bit of background on this drug. It's a derivative of a loop diuretic, and it's used to treat high blood pressure, and it's already approved for use, so the clinical safety as well as the efficacy has been shown. And research has shown that there's a robust inhibition of migration and invasion, as you can see left and right graph respectively. As the dose goes up, you see that both parameters go down significantly. And also, it induces apoptosis in the colon cancer model that was previously shown. And we want to extrapolate this to the ependymoma.
So how do we approach this? So the first noteworthy challenge would be the actual genetic variability of the tumor, its grade and location in the CNS. So actually there's a 20% chance of metastasis because of its location near the CSF pathways. And we realized that, as you see the two blue bars here, that it's very abundant in aquaporins 1 and 4. And aquaporin 1 is associated with cancer development, progression, and migration, whereas aquaporin 4 is associated with cerebrovascular diseases such as brain edema. And ultimately, what we want to do is we want to give this drug post-resection. So we resect the tumor, and we want to inject the drug at the site of the tumor itself to circumvent the blood-brain barrier and to localize the effect of the actual drug. And ultimately, you want to prevent it from recurring and to overall increase the event-free and overall survival of the patient.
So our hypothesis is actually quite simple. Instead of going with a traditional model of surgical resection and the chemoradiotherapy and then having the chance of recurrence and metastases, what we want to do post-surgical resection, we want to inject the inhibitor of the drug at the site of the tumor, and then what we hope will happen is that it will prevent the recurrence and metastases of the tumor, and it will ultimately lead the patient into remission.
So we broke down our approach into 3 aims. The first one is to explore the role of aquaporins 1 and 4 in vitro on patient-derived ependymoma cells, and we're going to use infratentorial ependymoma cell lines. And then we want to see the efficacy of the drug itself in reducing the proliferation, invasiveness, and migration of the actual cell lines. Then we want to test the ability of this drug to reduce angiogenesis in mice that are artificially affected and transfected with the infratentorial ependymoma. So to go about aim 1, what we're doing is we're going to create 2 models. One is an overexpression cell line, and another one is going to be a knockout cell line. So we're going to take the patient cell lines EPN-PFA and EPN-PFB. And in one batch of EPN, PFA, and PFB, we're going to retrovirally transduct them with aquaporins 1 and 4. And as you can see, you're going to have a lot more expression of the channels. Then in the other batch, we're going to actually knock it down with siRNA interference. And that way, they're going to have almost close to no expression of these channels. Then we're going to do a cell viability assay. So with this, we're going to measure EDU incorporation into their DNA, and so we'll just allow us to see what cells are viable, how they're doing now, if they're more proliferating, or if they're going apoptosis. And then we're going to do FACS cell counting to again sort the amount of alive cells versus dead cells, just to see how that affects their proliferation. Then we're actually going to do a wound healing assay. So we have the overexpression line, and then we have the knockout line. Then we're going to take the wild type PFA and PFB and have a treated group and an untreated group. Then in all those lines together, we're going to grow them on a confluent monolayer on the following substrates like collagen, fibronectin, laminin, and then we're going to do a scratch test. We're going to create a scratch, and then we're going to use DCIM every 4 hours to basically see how fast the actual scratch we created is covered up by the migration and invasion of the tumor cells.
Okay, for our third aim, what we're going to do is we're going to take a GFP transgenic mouse. This is going to be our in vivo model, and we're going to intracranially inject ependymoma stem cells that have been transfected with RFP. So you'll have a differential signal between the mouse cells and the ependymoma cells. Then we're going to have two populations, mice, a control mouse population and a treated mouse population. And the control one's going to receive just a saline injection at the site of the tumor, whereas the treated population is going to receive the drug AQB013 at the site of the tumor. And over the course of 60 days, what we're going to do, we're going to track the progression of the tumor using fluorescence imaging, and we're going to compare the invasion and migration of the tumor. Also, once we're done with this experiment, we could also sacrifice the mice and do tissue sections, to do histology, and to try to see if there's an effect on angiogenesis between the treated and the untreated population.
So what do we expect from this? In both the knockout and treated cells, because the drug treatment is synonymous with the knockout, because we're now impairing the aquaporin 1 and 4 function, we should see less migration and invasion in both those cases, compared to the overexpression line and the control DMSO line. Then, in terms of proliferation, we should see a lot less cells in the S phase, or G2 and M phase, in the knockout and aquaporin treated batches, compared to the overexpression and control batches, as you can see by the bar graphs as well.
So for our third aim, once we do the fluorescence imaging, you see that the control mouse, the tumor has actually grown in size and has metastasized, as you see by the RFP signal. And the actual treated mouse should have a more localized, smaller area of RFP where we injected the ependymoma stem cells. So there'll be a much larger signal for the RFP compared to in the control mouse compared to the treated mouse. At the same time, if we did the tissue sectioning and the histology, you would see much less microvascular density in the treated mouse compared to the control mouse, because aquaporin is connected to angiogenesis and spread of the tumor. So future directions, like with any drug, you need to do optimization toxicology once you do the in vivo model, and combination therapy has been found to be really useful, especially with other derivatives of the loop diuretic. They found to potentiate the inhibition of the aquaporin water channels, and we could also extrapolate this treatment to other tumors that are highly expressed in the aquaporin proteins, such as the colon cancer model that we saw, the adenocarcinoma model. We could also have it a targeted immunotherapy, but the problem with this is that aquaporin is actually a ubiquitous protein, so there could be pretty nasty side effects if we actually use immunotherapy. Lastly, and most importantly, we want to isolate the ependymoma stem cells, and we want to perform a single-cell RNA sequencing to get the gene expression profile and see which genes are actually differentially expressed. So we could do a pathway analysis to see which pathways and which enzymes are linked with the aquaporin's role in the pathogenesis of ependymoma. Thanks.
Thanks for your presentation. Are you, just to confirm, are you proposing injecting this AQB013 like intraoperatively, like immediately post?
Yeah, yeah, exactly.
Do you know of any studies or any evidence regarding the effect this this compound would have on normal brain tissue as opposed to the tumor?
No, we don't, because this hasn't been tried on brain tumors. It has only been tried in colon cancers and other urothelial cancers. So that is why we want to start with tissue culture, cell work, and then move on to in vivo models, and then hopefully study with histological sections to see how mice brain development has been altered, and then from there proceed on whether it's safe to move on to humans or not.
And what was the supportive evidence in the adenocarcinoma model? Like what were the outcomes they found or the side effects?
Well, in this particular model, they realized they did a similar tracking with the colon cancer markers, and they realized they metastasized much less. And also the tumor, after a longer period of time compared to the control batch, it did not invade as much. So it was more circumscribed and localized. So it's a potential therapeutic agent for colon cancer because it's an antidiuretic. However, we're not sure. It's kind of aberrantly expressed in the infratentorial ependymoma, which is kind of bizarre. So we want to explore that further.
Do you know the normal expression of Aquaporin 4 in the brain? Is that normally expressed in
Well,
abdomen?
it is expressed. It's expressed at the end feet of the astrocytes. So the reason why we're injecting it the localized at the site of the tumor after resection, so that we can have only a localized effect and it wouldn't have more of a systemic effect if we were, let's say, to do an intravenous administration. So we're trying to prevent any unnecessary side effects by injecting it at the site of the tumor.
Because Aquaporin 4 is actually a major player in
Yeah.
the blood-brain barrier. Right? Yeah.
So in Xavier's case, he had brain edema, and we see the infratentorial ependymoma here, and the Aquaporin 1 and 4 are much higher than the other areas. And so, as you can see also by the tissue section in the posterior fossa, so depending on, we're only looking at this particular subtype of ependymoma. We're not sure if it would be applicable to ependymomas in other areas like supratentorial or in spinal ependymoma.
Your assays seem to concentrate on invasion and metastasis. Is that right? You also mentioned angiogenesis. How are you going to measure that?
We're thinking of doing tissue sections after we sacrifice the mice, and from there we can do a microvascular density on the slides, or we can do fMRI of the actual mice itself to see whether metabolic rate has increased in the tumors, which could be inducive of more vasculature and more nutrients, and also blood levels of like VEGF and other markers of angiogenesis.
So you in an ideal world, you'd like to image the mice. Is that what you're saying?
Yes. Yeah.
How do you think